Thanks a lot, Robert, for the introduction. So it's a great pleasure to present my PhD topic, which is involved in transposable elements or TEs, and particularly with a focus on the silencing by piRNA clusters. But before we go to the specific topic on piRNA clusters, let me start with a short general background on TEs, and particularly their contribution to genome size is substantial. And this also varies across species, so like ranging from yeast of about 5% of transposable elements up to more than 80% in maize. There is a large variation in TE content. And these organisms have to deal with this large amount of TEs, and TEs are generally, when they active, they are generally known to cause damage to the host because due to insertions, they can mutate coding sequence when they insert in, or even if they are just closely inserted to an expressed gene, it can affect its gene expression, but also it can induce large-scale genomic instability or rearrangements by producing producing double-strand breaks due to its activity, or also ectopic recombination between homologous sequences of TEs. So the host needs to control their activity, and it does it mainly on the epigenetic level. Oh, sorry. And here you can see the main silencing mechanisms that involve, for example, histone modifications that repress the expression, or changing the chromatin structure of the TE region, but also directly methylating the DNA can silence the TEs. And importantly, there are small RNAs, siRNAs and piRNAs particularly, that allow to target specifically TE sequences to nascent RNA at the expression site to put repressive histone marks or methylation, as well as post-transcriptionally, TEs can get silenced by piRNAs, and where the TE fragment gets cleaved. And let's focus on the Pyrinate pathway, because this is the main mechanism in Drosophila where we work with. And you can see that here a scheme of the Pyrinate pathway that is functional in the germline. And here you can see in brown TE sequence that if it gets expressed and you have the nascent RNA here, the piRNAs, as shown here by this purple line, bound to, for example, PW and other proteins are bound there. They can induce here repression of the expression by depositing repressive histone marks like H3K9 methylation. But even if the TE makes it outside of the nucleus as a mRNA to express its own proteins that are encoded here that it needs for its transposition activity, the host can here cleave with, for example, OP that is bound to the antisense piRNAs, the TE mRNA into smaller fragments. And these fragments can be even used then by the piRNA machinery to cleave further piRNA precursors, and by this enhance the response to this TE in a positive feedback loop, the so-called ping-pong cycle. And this can feed also back to the nucleus here to silence the transcription already. And let's focus on these piRNA precursors that originate from certain loci that are the source loci for the piRNAs. These source loci are also called piRNA clusters, as they frequently contain many different fragments of TEs that can give rise to different piRNAs. And these piRNA clusters, they are located between the euchromatin and the heterochromatin, and they are believed to act as traps since they produced most of the piRNAs. And you can see here also a IGV track from such a cluster, that you have many different TEs here sitting, and if another T jumps here inside, the host can now produce also RNAs against this new insertion and silence all other copies in the genome. And this observation gave rise to the so-called trap model in the silencing of transposable elements. And this will be important later on. I will come back to this trap model. Let's focus maybe on the broader aspect of the project, where we focused basically on two aspects. One is the evolutionary dynamics, which is poorly described. And so because these piRNA clusters are so repetitive, until recently they were not really accessible by sequencing techniques of short reads. And the second aspect is that it's still not clear to which extent the piRNA clusters contribute to the silencing, since there is contradicting literature. All right. And now, this is generally the outline of the presentation, where I split this presentation into three topics. The first one deals with the technical aspects of analyzing piRNA clusters, while the second one approaches the biological question on the evolutionary dynamics of piRNA clusters, and the third one will try to test the trap model. All right, so let's start with the technical part. We can analyze piRNA clusters on the with genome assemblies, so with the assemblies of piRNA clusters from different lines or populations. And as I said, it wasn't really accessible to short reads, but with the help of long reads, we can assemble piRNA clusters more reliably. However, even if we do this, we don't know how to identify the correct piRNA clusters. And so we approach this in this project by establishing a new quality metrics to assess the quality of piRNA cluster assemblies. And later on, we will approach the question on how to analyze these repetitive regions. And the quality metrics that we established, we did together with Florian Schwarz, a former PhD student in Robert's lab. And the first metric we invented, we called Cusco, in analogy to Busco, which is a frequently used metric in assessing the genic parts of genome assemblies. And for this, we rely on unique flanks that, since the internal part of the clusters and not the conservation of the internal part of clusters is not well known, we relied on unique flanks that we use to identify the clusters. And you can see a cartoon for this here. So we have in our reference genome, we have the piRNA cluster annotations that are already available, and we could obtain the flanking sequences here on both ends, and then use them to align it to other genome assemblies of different Drosophila lines. And if we find both flanks on the same contig or scaffold, we can assess that this cluster is complete. However, we can still distinguish between really gapless assembled clusters like here, or clusters that still have missing sequence but just got scaffolded due to in the assembly pipeline. But next, it could still be that we have some misassemblies here, even in a gapless assembled piRNA cluster. So we approach this problem with two further matrices that use the long read alignment of those reads that were actually used to assemble these long read assemblies. And there we look for two patterns, namely the coverage of the long read alignment, as well as the soft clipped reads that align to the clusters. Soft clipped reads are reads that do not fully align to a query, but some part is not able to align, and this could suggest also misassemblies. And we compare these patterns in clusters against patterns in complete Busco genes of the assembly. And you can see a cartoon for the coverage quality here. So in a correctly assembled cluster with two repeats here, shown in blue, we expect a more or less homogeneous coverage. However, if we are missing one of such a repeat, then we expect here a dip in the coverage or a peak at the other repeat. And you can see here two real examples of one correctly assembled cluster and one wrongly assembled cluster, where the dotted lines are the 99% quantile range of the Busco genes. And you can see that here we indeed had a repeat collapse in this region, and so we could spot this with the coverage fluctuation here. And here the soft clip quality, if we have a correctly assembled cluster, then we ideally expect no soft clipped reads here, as all reads can fully align here, or at least little soft clipped reads. But if we have a wrongly assembled cluster, we again expect a signal of the reads. And so here again, the same assemblies. Again, we can see here the problem here by this peak from the soft clips. So after establishing our matrices, we could now start to find the clusters and look at their quality. And you have here an example of a famous cluster 42AB that produces a lot of piRNAs in melanogaster. And in the middle, you have the reference piRNA cluster of the reference strain ISO1, and two pairwise comparisons with two other assemblies of two other Drosophila melanogaster lines that we assembled. And you can see here the nucleotide alignment with BLAST. So these blocks are from BLAST. And we can see that most of the sequence aligns. However, there's also some variation between these three lines. And we can now take our two other matrices, the soft clipped and the coverage quality, to look if these regions are nicely assembled. And in Canton S, indeed, the signal, it gives a good result, suggesting a good quality. However, in the Pi2 strain, we have several peaks, especially for the soft clip metric, like here in the middle. But here, these are simple repeats. So, and we couldn't find any other repeats that could be assigned to these peaks. However, here in the end, we have another peak. This one we could assign to a TE. So this was actually the P element, and this was missing from our assembly. So it didn't make it through the assembly pipeline. And like this, we can find our potential misassemblies. And another point that I want to make is that you can see that here with the nucleotide alignment from BLAST, we have difficulties to interpret these results, as we have matches also between different repeats across the cluster, making it difficult to analyze which TEs are fixed or which TEs are polymorphic. And therefore, we thought we need to find another way of analyzing these regions. And so, inspired by the protein sequence alignment, where you align amino acid instead of the underlying nucleotide sequence, we align TE annotations of the PyRNA clusters. And so this is a multiple alignment of TE annotations, which we call MANA. And in brief, MANA employs the Needleman-Wunsch algorithm for a global alignment of the sequences of TE annotations. So each color is a TE. And like this, we can easily find polymorphic sites, like here, of insertions or deletions, or also segmental duplications. And this allows us now to approach the biological questions, like the dynamics of the PyRNA classes. And this brings me to the second part, where we did a collaboration with Sarah Signor, who is also interested in PyRNA cluster dynamics. And we used to address this question, we used four different Drosophila strains. So Drosophila seychellia, simulans, mauritiana, and our reference species Melanogaster, to look at variation between species, but also within species of Melanogaster and simulans. We looked further also within simulans into a population where we had two assemblies of the same population, and like this we could infer the dynamics of these regions. So we used our Cusco approach, that is here in gray, where we used our flanking sequences from Cusco to find also the clusters in other lines of Melanogaster, but also lines of other species. And then in the next step, to validate the orthology, so we are sure that we are comparing the orthologous regions, we designed further flanks in the target species and aligned it back to our reference species. Like this, we could make sure that it's really the orthologous sequence. And you can see here an overview of the set of clusters that we could use. So, of course, in Melanogaster, and these are only ungapped clusters. So all the gapped clusters we discarded here. And of course, in Melanogaster, we found the most clusters, and the gray bars also showed that those that are only specific to this species, that we couldn't find gapless in the other species. And also some intersects, like here, the three Melanogaster have, of course, the most clusters shared that we could find in all the three assemblies. But also in the other species, we could find decent amounts of our clusters. And particularly, we focused here on these 20 clusters that are shared between the 8 assemblies that we did the most analysis with, and those we validated with our orthology approach. And then also for small analysis, we used also the second assembly of the same population here, the SC45, where we had less clusters assembled, gapless, and had only a subset of these 20, namely 16. And with this, we analyzed the dynamics. And this brings me to the first result now, where we compared the divergence between species. Okay. Where we took just here for an illustration, you see for the famous cluster 42AB, just really for visual purposes, we see that there is really a high divergence between the species, although less so in the very closely related ones here. But to Melanogaster, there is really a large divergence. And you can see here the corresponding phylogenetic tree of cluster 42AB, also including the lines of Melanogaster and the lines of simulans. And also this pattern seems to be general, as we see that the average tree across all the 20 classes looks pretty similar in the sense of how diverged these clusters are. As here, this line represents a distance of 0.2, which means that 20% could not be aligned between the samples. Okay, and we also complemented our analysis with an alignment-independent approach, where we just looked which TEs are present in which clusters. So we have the TEs here in the rows, and in the columns we have the clusters of 3 different comparisons, namely simulans and melanogaster, simulans and mauritiana, and simulans seychellia. And we see in the green dots are marked those clusters where we have the same TE in the same cluster. And however, most of the TE content is private to one of the species, as you can see here from the numbers, especially in the comparison between simulans and melanogaster. Really the very large fraction is private to one species. But also in the closely related species, a substantial amount is private to one species. So this suggests that even with our alignment-independent approach, just comparing the content, there is a high turnover within these regions. And here we will start to focus more on within species comparison, where we had 3 assemblies for simulans and 3 assemblies for melanogaster. Again, our famous example of cluster 42AB. We see that within the species, most of the cluster is really very similar, although there are some polymorphisms. And we can see that also in general for all the 20 clusters here from the site frequency spectrum, where we have most sites fixed, but still there is substantial amount of singletons and doubletons as well. And what we can see as well is here, if we look at the absolute counts of T's in the site frequency spectrum, we see that the melanogaster clusters have much more TEs than the simulans. And this can be also seen here. So it's not just that they are longer, but they have really more TEs here. And but if we look, for example, on the individual clusters, we see also that these clusters are evolving at different rates. So in the transparent color, we have the fixed sites, and in the opaque color, we have the polymorphic sites. And while for most clusters, there are more fixed sites, as already seen in the site frequency spectrum, some clusters have more polymorphic sites in the species, like here, or here, both species have roughly the same amount of fixed and polymorphic sites. Also, simulans has, for example, cluster 5, which has way more polymorphic sites than fixed ones. So this suggests that the clusters might evolve at different rates, also, which is also species specific. And in the next step, we now include this 4th simulans assembly, which was from the same population here. And we found there a striking pattern, namely there are polymorphisms like in this example, this max insertion that is segregating within the species. However, this polymorphism can be also found within the population. So we checked if this is general for all our clusters by pairwise comparisons and counting polymorphic and fixed sites. And you can see that the pairwise comparison within the population shows a very similar level as other pairwise comparisons. So this suggests that indeed the variation within a population could be similar to the species variation. And in the last part of this sub-project, we looked at how the clusters evolve. So if there are, particularly we looked at insertions and deletions, and if there is a bias. And indeed, we found a bias for insertions. So the clusters are actually growing and acquiring insertions. I have to mention that we did this only in simulants, as we could only use, as we needed the outgroup, and this was only possible with the closely related species, as Melanogaster was too far diverged. So we used here Mauritiana as an outgroup to infer the ancestral state, and then we found that insertions are dominating and over deletions, and also that they are longer than the deletions. And furthermore, if we looked which families, which TE families are involved in insertions or deletions, we found that insertions involve families that have a lower population frequency, and which is a proxy for the age or the time of activity. So if we have lower population frequency of TEs, this means that they were recently active or maybe are still active. And so by this we could infer that the clusters evolve through insertions of young TEs and slow degradation by deletion of smaller fragments. So to conclude on this is that we found that the clusters are evolving extremely fast and have a substantial variation within the species, but probably also within a population. And further, they evolve by insertions of young elements. All right, and this brings me to the third part already, where we try to test the trap model and if the clusters are really responsible for the silencing of the TEs. And let me elaborate a bit on the trap model, which assumes that a TE, an active TE, can jump in the genome and acquire higher copy numbers until it hits randomly a PRNA cluster, and then the host can start to produce PRNAs that silence all other copies in the genome and thereby inactivates the TE. And so we can perform simulations of TE invasions under the trap model. And you can see such a simulation result here, where we simulated 300 replicates, these lines here, of TE invasions. And this is, you can see in the beginning, a rapid increase in the green phase where there are no PRNA cluster insertions. But as soon as some TEs jump into clusters, we get the curve starts to bend, and at some point we have a plateauing of the trajectory, and then eventually the fixation of cluster insertions that is shown then in red. And interestingly, this one cluster insertion was not enough. So in actually to really silence, to get into the plateauing phase, we see that we need about 5 to 6 cluster insertions. So you can see this here in the distribution of cluster insertions, so the count of cluster insertions and the amount of the fraction of individuals carrying the certain count of cluster insertions. And we see that at this phase where we have the black line, we expect about 5 to 6 cluster insertions, and the distribution of cluster insertions should be more or less symmetrical around this value. While for some random reference regions in the genome, some control regions, this should be more skewed, as we see here. And now we can check our melanogaster lines, how the distribution of cluster insertions or reference insertion look like there. And here you can see again the expectation of cluster insertions where we have in red the 98% quantile range of all the individuals here, and here the observation in the real data in our melanogaster lines, and again with the quantiles based on the expectations, we see that the distribution looks very different from our expectation, where we have substantial amounts of individuals without certain cluster insertions, or also individuals with many cluster insertions. So an overabundance of cluster insertions. And this is our hint that maybe the trap model is not really applicable to the silencing of TEs. Another feature that we can use to compare simulations against real data is the TE abundance. So so we can correlate the abundance of TEs in pyRNA clusters versus the abundance of TEs in the rest of the genome. And the same we can do also for some random control regions. And while for the random control regions, we expect a strong, more or less strong positive correlation, because these are just some random regions from the genome. This is not the case for pyRNA clusters, where we rather expect no correlation or even a negative one. And we tested this again in our lines. Here you can see in the top the cluster correlation, and in the bottom the correlation with reference regions. And actually both have positive correlations. So actually the cluster regions look very similar to some reference regions in the genome, and this was not expected under our trap model. And this brings me to our conclusion before I allow myself to speculate a bit on that result. So we think that the simple trap model likely does not explain the silencing of TEs, since we have many missing cluster insertions, as well as an overabundance of cluster insertions. And this lets me speculate that maybe there was persistent activity even in the presence of sufficient cluster insertions. And we can also find hints that this is really the case as we look for insertions, really de novo insertions that we can find since they are really restricted only to one particular TE and not larger fragments of the cluster. We can find sometimes the same element sitting in the same cluster several times, and it looks really like a de novo insertion and not maybe a repeat expansion, which is also the case. And this repeat expansions we also observe, and they could be also responsible for the overabundance that we observe. Like here you can see some array of copia and Quasimodo that is triplicated here, as also supported here by these larger synteny blocks. So this could partially explain our overabundance, but still we had these cases of de novo insertions that is clearly not expected on the trap model. And this let me speculate further that maybe these repeat expansions are also relevant to the cluster evolution and maybe to the silencing of TEs. But this leaves an open question. And actually, with this open question, I would like to end the talk, and thank you a lot for your attention. I would like particularly thank my advisor, Robert, for the fantastic support, and also my other two advisors, Joachim and Kirsten. as well as the collaborators of this work, Florian and Sarah, and the graduate school for this great opportunity, and the Institute for support and feedback. And yeah, so I'm happy to take questions.